Hi, I'm Peter Huang, and I'm the CEO of Marker Therapeutics. We're a publicly traded company on the NASDAQ, and so we always have to start out with our forward-looking statements, disclaimers. I will be making forward-looking statements, and I encourage you to look at our public filings for a comprehensive description of all of our risk factors. So we have a very different and I think novel approach to cell therapies versus the typical CAR-modified or TCR-modified T cells or even a TIL program, in that we have a program where we are generating a T cell population that is not genetically modified but is selected to be tumor-specific to multiple tumor-associated antigens. In fact, there are many differences with this approach. The first being that we tend to use a much lower dose of cells. So our dosing range currently ranges from 5 million cells per meter squared to about 20 million cells per meter squared. That is roughly translated into a large adult, no more than 40 million cells a patient, which, as you all know, is a comparably very low dose of cells. In addition, we currently do not lymphodeplete the patient, and so we do not use cyclophosphamide or fludarabine prior to using our therapy. And like I said, we do not gene modify the cells, and so this all allows us to produce a product at considerably lower cost than a typical virus or transposon modified cell therapy program. So versus a traditional CAR-T or TCR therapy, I apologize here. I seem to be having some trouble moving the page. Oh, here we go.
Is it animation or?
There is an animation. A typical gene-modified cell program, as you know, targets a single antigen, although we know that many tumors are heterogeneous in nature. And so when this program was initiated at Baylor in 2012, the goal of the program was to try to find a way to address a very heterogeneous tumor by creating a product that was equally heterogeneous. And so when we talk about the program itself, it looks like it's working on a lag with me here, so bear with me through the animations. Oh, look at that. It looks like I've far over-clicked. So.
Return everything.
Sorry.
Or is over-clicked, and then you click back, so it's now going forwards and backwards. Have animation. You were on a slide number 6, 7?
7. This one, you can put it on here. Thank you very much.
Absolutely.
So ignoring the technical difficulties here, the question that I think that is natural to ask is, look, I mean, if we're not lymphodepleting and we're not genetically modifying and enhancing the T cells, and that we're using a very low dose of cells, how is it that we expect that we'll be able to get therapeutic results? And so if you look at the bottom part of the page here, what I would tell you is that what we're finding in our clinical results is that I think that our T cells, this cell therapy is acting very differently than a typical cell therapy, in that cell therapies today are generally primarily focused towards direct tumor lysis. That is that the therapeutic effect is driven entirely by the direct killing of the infused T cells. And what we're finding in our program is that our cell therapy is acting in a way that's very different. In fact, it's more like a hybrid of a traditional cell therapy and something of a checkpoint, in that our infused cells do initiate a process of direct tumor lysis, exposing antigens, and helping to convert a previously immunosuppressive environment into one that's capable of recruiting the endogenous immune system. And secondly, And we think that this effect may have even greater effect than the direct tumor lysis caused by our T cells, our infused T cells, acting like a checkpoint in terms of re-engaging the endogenous immune system. So when we introduce a multi-antigen specific product, in this case specific for up to 5 tumor-associated antigens, we can do broad-based killing up front that exposes target antigens and helps to recruit the native endogenous immunity to attack those tumor cells that express even antigens that are not included within our product itself. And so how do we do this? So today the manufacturing process is identical for all of our products, although there is a difference in the source of blood. So to the extent that a patient is getting an allogeneic stem cell transplant, we always want the T cells to match the stem cell repertoire of that patient. And so if a patient is getting an allo transplant, we take blood from the donor. If a patient is not getting an allo transplant, then we draw blood from the patient. To date, because we've required so little blood, in fact, we can manufacture with as little as 80 mils of blood. We have typically manufactured from blood drawn by needle draw rather than by apheresis. And the manufacturing process itself is extremely simple. So we separate the blood into monocytes and PBMCs. Up front, there's a 7-day maturation process in order to mature those dendritic those monocytes into dendritic cells. Once the dendritic cells are matured, then we simply put them in a cell culture device with an aliquot of the PBMCs, a cytokine mix that has been designed to prevent antigenic competition. That is, that typically what people found was that it was possible to grow T-cell monocultures based on a single peptide, but to the extent that you tried to grow a polyclonal T-cell population in a monoculture, you'd find that the most immunogenic T-cells would invariably push out the lesser immunogenic T-cells. And so the cytokine cocktail that we use is important in terms of preventing that competition and growing as close to a 1-for-1-for-1 repertoire by T-cell clonality as we can get. In addition, we add cytokines, growth media, and then we just let it sit. And in this cell culture device, what we're trying to do is to recapitulate the internal antigen presenting process, effectively to create an ex vivo lymph node. And the cell culturing process here helps bring T cells that were previously highly energized and highly tolerized back to function. And we can show that in that, if we isolate T cells that are directly withdrawn from peripheral blood and we run an overnight cytotoxicity assay, you'll find that those T cells are non-functional, and that this cytokine culturing process is important for restoring these T cells back to function. We do 2 to 3 stimulations that are identical here, and we just let it sit. At the end of it, we wash, cryopreserve, and test the cells, and then we ship them to the site. Because we're using such low doses of cells, the infusion is actually a 2 ml infusion that is done in less than 10 minutes. We monitor the patient for 2 hours for any infusion-related reaction, and then we send the patient home, because in 100 patients dosed to date, we have never seen a case of CRS or a severe adverse event. In fact, in 100 patients dosed to date, we've only seen 1 grade 3 adverse event that was deemed to be related to our T cells. I'll tell you about that patient later, but that event may actually have been a false positive. So one of the important things about this process is that they were undetectable, either by tetramer staining or by deep sequencing. That is, that they were so rare that they fell below the threshold of detectability. And it was only after the manufacturing process that these T cells were expanded to a scale that we could detect by tetramer staining or by deep sequencing. And we infer that they were expanded to a scale that would be necessary to generate a therapeutic effect. The second thing that was important about these T cells was that when we looked through all of the prior literature, we could find no disclosure of these particular TCR sequences. So as far as we know, these are previously undiscovered TCRs. We don't consider ourselves a target discovery company, but to date, our library of previously undiscovered TCRs, that if you believe the correlative data, mean that these TCRs are disproportionately important in driving an anti-tumor effect, now numbers in the hundreds. The last observation we made was maybe the most important, And when we assessed these three particular TCRs by affinity, trying to assess functional avidity, they were by no means the highest. And so had we tried to predictively generate these TCRs, we never would have guessed that these TCRs were important for driving an anti-tumor effect, being therapeutically relevant, and yet we see that a third of the anti-tumor effect, as measured by expansion of the T cells in the patient, comprise these particular clones. What I'll do is I'll actually skip to some of the clinical data to show that, that this therapy can be particularly relevant. So these are 15 patients who had active disease in lymphoma who were treated with our therapy. The blue lines here represent patients who got stable disease. The green lines represent patients who developed the CR. The red caps represent the time that those patients actually progressed. And the first thing that I'll note is that the worst we did for patients was that we saw transient stabilization of disease. And so when you look at the stable disease patients, you see that these patients have sometimes relapsed between 3 to 9 months out, although there's a notable example of a patient who seems to be indefinitely stabilized post-receipt of the T cells. The interesting thing about this for me is that when you look at the overall time to progression, it actually closely resembles the progression-free survival curve of all patients who received a CD19 CAR. Now, this patient population is more diverse than what we see in a typical CAR study, and that includes both Hodgkin's and non-Hodgkin's patients, but we have not been able to discern any meaningful difference between the patients who we treated with Hodgkin's lymphoma versus say, DLBCL. Of the DLBCL population, there were 7 patients with DLBCL. 4 of them generated CRs, although one of them was a little bit mystifying. That's patient number 15. That patient was confounding because they received our T cells in December of 2017. Saw a little bit of a decrease in SUV from double digits to 8, but we saw no resolution of the tumor by PET scan, so we scored that patient stable disease. The patient actually ended up progressing on to an experimental CD19 therapy in April of 2018, saw no response to that therapy, including no evidence of CAR-T expansion. Interestingly enough, when we actually did a deep biopsy of the patient in November of 2018 in preparation for ASH, that patient was found to be disease-free. So we know that that patient converted to CR sometime between December of 2017 and 2018. Whether that was a complete response to our T cells or the experimental CAR, it's impossible for us to say. But what I can say about these patients is that they are heavily pretreated patients. So rather than being closely controlled for 2 to 4 lines of prior therapy, these patients on average failed 4 to 7 lines of prior therapies. Several of them failed as many as 9 lines of prior therapy before coming onto our cells. I think it's possible to overstate the comparison with CAR-T, though, because I think that what's important to take away from this is that I think that this is a very, very different cell therapy in its own right, and that in the long term, I have a vision for this therapy where it may be used broadly as a maintenance therapy for patients who are in remission from any prior therapy. So because these cells appear to do very little harm to patients, because we can produce these cells relatively inexpensively and price the therapy at a level that's appropriate for a maintenance therapy, and because these cells are natural T cells, in the absence of tumor, we don't worry about them disappearing because we think that they retreat into memory phenotype. There's a school of thought that says that no different than providing a woman hormones after surgical resection after breast cancer, that any patient in remission from any prior therapy, including first-line standard of care, should just get cells as a matter of course if we think that these cells are protective. And we do. So these are patients who have received cells after generating a remission from a prior therapy. What I will tell you is that these patients were so refractory that their best prior remission for the first 11 patients ranged only from 1 month to 5 months, with a median of 4. You can see here with the length of time in ongoing complete remission, over 3 years, over 3, over 4, over 2, over 4, over 3, over 2, over 3, over 2, over 2, and over 1 year, that we can definitively say that we have generated the best ongoing remission for these patients while receiving these cells as a maintenance therapy. And these patients now, the first 12, have a median duration of CR that approaches 40 months. You should be seeing these patients at this point approaching their median progression-free survival, and you can see that we are nowhere near the median progressor. In fact, patient number 2 and number 3 are the same patient here, and so we've only seen 2 progressors. We have not seen a single progressor in the last 13 months. Patient number 2, by the way, is a patient who received cells after generating a CR to R-CHOP, saw 8 months in CR post-R-CHOP, relapsed, then got another CR from arbendastamin, received our cells again as a maintenance therapy, and is now over 3 years ongoing in complete remission with the use of the T cells alone as a maintenance therapy. So once again, why is this happening? Why with a small dose of T cells, no lymphodepletion, and no gene modification, are we seeing these results? And we believe that the answer is that that we're getting significant assistance from the re-engagement of the endogenous immune system. So you can see here, this particular patient, prior to infusion of our T cells, had very few T cells that were specific for the antigens that we infused. After we infused our T cells, you see an expansion of T cells that closely matches the antigen expression level on the tumor, being a high SSX2 and a high NY-ESO-1 expressor. But the reason that we think that this CR has been so durable, over 3 years now in ongoing CR, is that we see consistently in our patients a second wave of T cell expansion, of T cells that we did not infuse in the patient. In this case, we can identify a group of cells that have expanded that are specific for MAGE-A3 and cells that are specific for MAGE-C1. WT1 here is the negative control. WT1 is not on lymphoma, and so it is our way of showing that we are not expanding T cells that are not specific for the tumor. This is a patient with a mixed Hodgkin's and non-Hodgkin's DLBCL. And what's significant about this is that when we started dosing patients in 2012, we not only started at a really low dose, 5 million cells per meter squared, so no more than 10 million cells for an adult patient, but the FDA was significantly concerned about toxicity, and so they forced us to antigen escalate before dose escalating. So we had to dose 2 patients with a single antigen product, wait 6 weeks, dose 2 patients with a 2 antigen product. It took us almost a year and 8 patients to antigen escalate to the full product. Thankfully, this particular patient was a high expressor in both PRAME and SSX2, the 2 antigens that they received T cells for. But when you see expansion of the MAGE-A4 and NY-ESO-1 T cells, in this case, that's epitope spreading, because that was not included in the product. One thing that's important to understand is that the kinetics of this are very different from what you may be used to seeing in a gene-modified product, in that because we have such a low dose of cells and that we're not lymphodepleting, we don't see the same sort of peak expansion of T cells that is typical in a TCR or CAR-modified cell program. In fact, what we tend to see is a slow expansion of the T cells. They end up comprising between 2 to 4% of the circulating lymphocytes in the patient's circulating peripheral blood. The patients typically report feeling better almost immediately after an infusion. We generally wrote that off to we have been consistently reported now amongst almost 100 patients. They do become asymptomatic within a few weeks, and they see disease stability very quickly within that few-week period. But when they convert to CR, it generally happens a month to 3 months after infusion of the cells. In this case, it happens sometime between month 3 and month 9. And here, what I think is notable is that if you look at the expansion of the T cells at month 3, you can see that the expansion is highly PRAME and NY-ESO-1 mediated. And I think that, that I would contend that there would have been a significant chance of losing this patient had we only had a product that was specific for PRAME or NY-ESO-1, in that you can see that by month 9, the expansion profile has very much changed. In fact, the ESO1 and PRAME T cell populations are actually in decline. So what we think is happening is exactly what the evolutionary biology would lead us to expect, which is as we kill down the PRAME and NY-ESO1 expressing tumor, the antigen expression is changing on this tumor. That tumor which does not express PRAME and NY-ESO1, in this case primarily expressing MAGE-A4, is beginning to grow out. And thankfully, our product was able to adapt alongside that. We also have significant patient experience in AML, ALL, and multiple myeloma. These are patients with relapsed refractory disease post-transplant who otherwise have really a historical overall survival expectation of about 4.5 months. These are patients that we treat as a maintenance therapy after transplant. I'm going to point out a couple of interesting things. One is that we seem to be deriving significant therapeutic benefit. You see significant debulking of disease, restoration of normal hemopoiesis, but here is probably the most striking example of the interplay between cells and tumor. This patient is a 57-year-old female who we treated in the adjuvant arm of the disease. So for the first 60 days, we don't treat to get her out of the GVHD period. At day 60, we treated her with our cells, and she got the only grade 3 adverse event that we've ever observed. It was a grade 3 elevation of liver enzymes. The doctors gave her steroids to treat that condition and suppressed our T cells. When they did so, the patient got significant extramedullary relapse, which you can see here along the spine as well as in her jaw. When the liver enzyme subsided, the doctors withdrew the use of prednisone. Our T cells spontaneously bounced back, and you see that a month later, with the restoration of our T cells, those tumor lesions have been greatly reduced, almost eliminated. In fact, the doctors elected to give her decitabine to try to clear up the remaining tumor lesions, and ablated our T cells. And when that happened, rather than helping, it put her into a full relapse. At that point, we contacted the FDA, asked to move her to the active disease arm. They agreed. And so we gave her a second set of cells, identical to the first. We actually just pulled them out of the freezer. And when we did, it generated a CR for the patient that was durable for 13 months. That patient survived 2.5 years after frank relapse. Being mindful of the time here, I will say that I think equally interesting data in ALL and multiple myeloma. Please bear with us. We've just gotten through our pre-IND meeting for our first phase 2 trial, which we hope to be a seamless phase 2. That will be in post-transplant AML. We anticipate filing the IND for that clinical trial in the mid-3rd quarter, with the first patient enrolled in that trial before year end. Also in that time frame, in the middle of the 3rd quarter, we do anticipate releasing our first solid tumor data in pancreatic cancer, where I think we've seen some interesting results to date. Please bear with us. I think that we hope to show some real activity in a solid tumor come the July timeframe. Thank you all for your time, and if you have any questions, please feel free or grab me afterwards. Thank you.